Hey guys, Mr. Cox here, your second favorite teacher, right after your lazy history teacher. We're going to talk about blood type genetics. So open your notebooks to a blank page and title it Blood Type Genetics. The next thing you're going to do is draw this 16 box Punnett square. So if you're a junior and you took biology last year, after spring break, if you decided not to do any additional work, the Punnett squares might be new to you, but that's okay. I'm going to walk y'all guys through it. So draw this 16 box Punnett square 4x4. And we've talked about Punnett squares once this unit already. We talked about it with sickle cell anemia. Now, sickle cell anemia is coded for by one gene, which means your Punnett square was just 4x4. It's a very simple Punnett square. Well, blood type, if we're talking about A, B's, and O's, and positives and negatives, it is two genes. Therefore, you have 16 box Punnett square called a dihybrid Punnett square. And this shouldn't scare you. I'm going to help you set this Punnett square up. And even on the test, I will help you set this Punnett square up. So all you have to do is know how to fill out the Punnett square and how to decipher the Punnett square. Neither one of those are particularly difficult. All right, so you got your notebook page titled. You got this Punnett square done. Let's take a look at this scenario. Okay, this is one scenario. We have a parent who this is their genotype, and another parent who's this is their genotype. Well, what blood type are they? We'll take a look at that first. So you'll have this little decoder thing when you take the quiz and on your study guide and stuff like that. So one parent is, we'll call this biological dad. Biological dad, we can see is type B. Is he positive or negative? Well, let's take a look. He has one dominant allele for the Rh gene. So, yep, he's Rh positive. So, biological dad is B positive. Let's take a look at biological mom. Biological mom is OO, so she's type O. And, yep, she has a dominant Rh positive allele. So, she's Rh positive. Now, some of these words you may not understand, like allele. Allele is just simply a kind of a copy of a gene that you have on one strand of DNA. You know, you get one strand of DNA from biological mom, one strand of DNA from biological dad. So you have two copies of the same gene. That doesn't mean they're identical copies. We can see here that these are two different copies. Now, biological mom in this scenario does have two identical copies. So she is type O. There's two ways to be type A. You can either have both dominant alleles or just one and be a carrier for type O. And to be type B, you can also have both dominant alleles or be a carrier for type O and still be type B. So we know that AB is not really a common blood type just because there's only one way you can get there. Type O is actually a common blood type. Why that is, I'm not quite exactly sure because it is recessive, but still about 40% of the population are type O. Now, O negative, having all four recessive alleles, that's quite rare. All right, so let's get started setting up this Punnett square. It's kind of like the FOIL method in math. You want to match each allele of the first gene with each allele of the second gene. The A, B's, and O's is a gene, and the positives and negatives is a gene. Now, what we're doing here, if we're basically turning the genes into what they would look like in sperm and egg cells. In sperm and egg cells, if you remember from bio, if you had bio after spring break, have half as much DNA as a normal cell. So we have to cut this DNA in half, but we have to do it randomly. So this part I will do for you on the test. We go ahead and write this down. So there's all the possible sperm combinations of biological dad with respect to these blood type genes anyways. And then let's look at biological mom. Let's make all the egg combinations. Now, she has two of the same type of alleles. So she has a little bit less genetic diversity with respect to blood type. All right, so there's all my egg combinations. There's all my sperm combinations. And what we're going to do here is we're going to bring these egg and sperm combinations, all 16 possible combinations, and those will be my offspring. And each one of these boxes represents a 1 in 16 chance of the child having that blood type. So let's do the first one together. We keep our A, B's, and O's together, and we put the dominant gene first. So it would be BO, even though the O actually is over here to the left. It doesn't matter. Dominant one goes first. And then capital D, capital D. Okay? So we know for a fact right off the bat, 1 in 16 chance that the kid's going to be type B positive, because 2 capital D's is Rh positive. So go ahead and write this out. Don't get lazy on this, especially on the test. So I'm going to say this first kid is B positive. Let's keep going down the list. I'm putting the B first before the O here. And I'm putting the capital D first. Now, what blood type is this? This, even though it's a different set of alleles, is still B positive because being Rh positive is dominant to being Rh negative. That's why there's more Rh positive people in the world than there are Rh negative, like 90% versus 10%. So this one's B positive. Now this kid will be a carrier for the Rh negative gene, but doesn't matter. His blood type is B positive. Here we have OO capital D capital D. So what's this kid going to be? O positive, the most common blood type. Next kid, OO. So once again, O there, O from there, capital D first, because dominant genes go first. And this kid's still O positive, most common blood type. Okay, I'll do a couple more of these for you, and I want you to go and do the rest of these yourself. I'll do the bottom row. If you don't know how to do this would be a good time to come to the Zoom meeting next week before the test. Okay, let's take a look at this bottom row's blood type. Here we have B positive, once again, BO, that codes for type B, and then capital D lowercase d, that codes for Rh positive, so B positive. Here we have, interesting, a B negative, so at least one of the kids is going to be B negative. Here we have O positive, and then here we have O negative. So the second rarest blood type, the universal donor, at least one of their kids is going to be O negative. I'm going to go ahead and do the rest, but once again, guys, I'm not going to be there to do this for you on the test, so make sure you understand how this is done. Yep, I got another O negative person here. O positive, O positive. B negative. All right, now, this is what you should have when it's all said and done. Not only should I have all my genes lined out, but what blood type that actually corresponds to. Now, we can tell this half of the chart is type O, this half of the chart is type B. So that kind of makes sense. One parent was type B, one parent was type O, and half and half of the kids. So they got a 50% chance of having a kid that's type O, 50% chance of having a kid that's type B. That actually makes perfect sense. Now, we need to look at this bottom one. What percent or fraction of the following phenotypes or blood types will have the following phenotypes or blood types? So what percent is going to be A, 0? We're actually going to do these out of fractions, so 0 out of 16, but 0 out of 16 is the same as 0. How many B negative? Well, let's count them. Let's make sure we go through all of these. 1, 2. So 2 B negative. So 2 out of 16 kids will have B negative. So if this couple had 16 kids, you could surmise that they would have at least 2 that are B negative. Now, we generally don't reduce fractions for Punnett squares. You could turn this into a percent if you were really on the ball, which I'm not. So let's move on. B positive. I got 4 there and 1, 2 there. So 6 out of 16 are going to be B positive. The reason you don't reduce it is because you could have a 3 out of 16, which would be would make your 1 out of 8 versus 3/16. It's kind of confusing, so we just leave it like that. AB negative, that's none. AB positive, none. But we do have some type O's. So how many O negatives? We got 1, 2. So 2 out of 16. And then 1, 2, 3, 4, 5, 6. So 6 out of 16. On the test, these should add up to 16. So there's 8 plus 8, that's 16. So we know we did it right. And then the last question is, what percent or fraction will be universal donors? Well, you have to ask yourself, what is a universal donor? Look back at this chart. We know that the only blood type that doesn't have any important antigens is type O negative. So the universal donor is O negative. So I look back at this, and 2 out of 16. So 2 out of 16 of this couple's kids will be universal donors. So their blood will be, sought after. Anybody can take it. All right, guys. So that is blood type genetics. You'll have another practice at this on the study guide before the actual test. Thanks, guys, and have a good day.